Welcome to Animated Science, Biology. This time we're looking at the electron transport chain, which is going to be creating most of the ATP our cells need to function. This is the reason why we breathe, eat, sleep. This is the reason why we live today, is because of this process in our cells and the creation of ATP. So electron transport chain is the fourth step of aerobic respiration. A quick review over the first couple steps. Glycolysis. It took place in the cytosol of the cell, and mainly that step split glucose and used some of the energy stored in glucose to create two ATP molecules, two net ATP, and also two NADH molecules, which we will utilize in the electron transport chain today. So after glycolysis, we have two net ATP and two NADHs per glucose. Moving into the next few steps, we moved into the mitochondria, which we looked at some of these layers. If you need a refresher on that, please click the card in the top. In the matrix, we looked at pyruvate oxidation, which took our two pyruvate molecules and split them up, and we created another NADH per pyruvate. So after pyruvate oxidation per glucose, that brings us to 2 ATP, 4 NADHs, and 2 CO2. The third step is the citric acid cycle. The citric acid cycle is going to create a bunch more of these NADH molecules, and also we created some of these FADH molecules. So these are electron carriers that we're going to utilize in electron transport chain. So these 10 NADH and 2 FADH2 molecules are going to be carrying their electrons to the electron transport chain. So the electron transport chain is going to take place mostly on the inner membrane of the mitochondria. So before we get into the details, we first need to look at oxygen. And now, if you recall from my citric acid cycle pyruvate oxidation video, that I said oxygen was required for the entire aerobic respiration pathway to occur. And so pyruvate wouldn't even be funneled into the mitochondria if oxygen wasn't here at this specific point in the electron transport chain. And so we're going to use a combination of a series of redox reactions. That's what the electron transport chain is. And oxygen is going to complete the last redox reaction. And it's the reason for this is because of its electron affinity. So if we look at the electron affinity chart, we can see that oxygen has one of the highest electron affinities, and so it's going to be the driving force for everything that we talk about in a second. So the actual electron chain itself is going to utilize 6 important enzymes. Of them, there are 4 that are embedded in the membrane. It's complex 1, complex 2, complex 3, and complex 4. We also have 2 mobile carriers, one of which is Ubiquinone, abbreviated with the letter Q, and that moves between 1 and 3, and 2 and 3, and cytochrome C, which is abbreviated as Cyt C, and that moves between complex 3 and complex 4. So 3 of these enzymes, complex 1, complex 3, and complex 4, are actually going to be hydrogen ion pumps. And they're going to create a situation in which the intermembrane space is higher concentration of hydrogen ions than the matrix. And so that's a really important thing to understand and that you need to learn. This is one thing that you need to remember. The intermembrane space is at a higher concentration of hydrogen ions, and the matrix is at a lower concentration of hydrogen ions. And the reason for that is because of these 3 enzymes that are pumping the hydrogen ions. And so if you need a refresher on pumps and membrane transport, I'll link a card in the top right. Go check out active transport. So one thing about active transport is we need some energy to power these pumps. And so in a minute, we're going to see how these 3 pumps are powered, because the high concentration of hydrogen ions is going to be a major factor that drives ATP synthesis in our 7th enzyme at the end of the video. So there's a lot of movement taking place, and we are going to zoom in and slow this process down, starting with complex 1. So the NADHs that were created by the previous processes is going to be oxidized by complex 1, specifically multiple cofactors within this. This iron sulfur cluster actually has a higher affinity for the electron than the NADH. So remember, we talked about electron affinity. But one thing that we should notice is that when the first cofactor, the first iron sulfur cluster, oxidizes NADHs and picks up an electron, there's a burst of energy that occurs. And so let's look at a little model to see where this energy is coming from. So if we look at oxygen and fluoride, and if we attach an electron to oxygen, let's say we reduce oxygen, we actually can observe an energy release. And now this is going to go down to the quantum level, so we're not going to explain why this occurs. It mainly has to do with the electron excitability. So oxygen, due to its electron affinity, releases 141 kilojoules per mole, and if we reduce fluoride, then it releases 328 kilojoules per mole. And so again, that goes back to the electronegativity scale that we looked at, which is a measure of the energy release when we do add an electron to the outer shell of these elements. And so for molecules, depending on the type of molecular structure and everything, they have their own electron affinity. And so complex 1 is going to have multiple iron sulfur clusters and cofactors that's going to be ordered from low affinity to high affinity. And so we're going to be transporting that electron through, and that energy is going to power hydrogens through complex 1. So as we see these electrons jumping from cofactor to cofactor, the electrons finally are picked up by ubiquinone, which is the enzyme with the Q here. And ubiquinone is going to transport them from complex 1 to complex 3. Okay, it doesn't go in order here. So from 1 to 3. Complex 3 is going to pick up the electrons. They're going to be passed through cofactors within complex 3. Not really too important here at this level to know all of the cofactors. And it's going to pass it to cytochrome C. Cytochrome C is going to pass these two to complex 4. And complex 4 is going to carry them to oxygen, which is the final electron acceptor. So oxygen is driving this entire process. If oxygen was not here, everything that happened previously would not have happened, all the way to the point the Krebs cycle wouldn't have happened, pyruvate oxidation would not have happened, pyruvate wouldn't even be funneled into the membrane. But once we add these electrons to oxygen, we're going to create H2O. And so let's back up for a second. We're going to go back to complex 3, ubiquinone protein. Once it's done here, instead of going back to complex 1, it goes to complex 2 to pick up electrons from FADH2. And its electrons are going to pass through cofactors of complex 2 and be passed to ubiquinone. Ubiquinone again carries these electrons to complex 3. Cytochrome C again delivers these electrons to complex 4. And complex 4 is going to deliver these final electrons to the second half of the oxygen molecule, because remember, oxygen gas comes in the form O2. Some of your textbooks may do some weird stuff and say that we have 1/2 O2. Basically, we take O2, we split the molecule in half. Complex 4 is going to be doing most of this work. Alright, so there's the electron transport chain. We powered some hydrogen ion pumps, but where is our ATP? We'll get there. So just a quick review over that. NADH is going to go take its electrons. The electrons are going to pass from complex 1. They're carried by ubiquinone to complex 3. Cytochrome Z picks up those electrons, carries them to complex 4, and finally delivers it to oxygen, which is the final electron acceptor. Now the FADH pathway, FADH2 delivers its electrons to complex 2. Ubiquinone is going to pick up those electrons and carry that to complex 3. Cytochrome Z picks up those electrons, delivers it to complex 4. And finally, we deliver it to the final electron acceptor, which is oxygen, to create our water molecule. Alright, so now we need to take a look at our 7th enzyme, which is ATP synthase. This is the enzyme that synthesizes ATP, hence the name. And so this is a fascinating enzyme which is going to have a rotor which turns. It's going to use the process of chemiosmosis. So remember, we had a high concentration in the intermembrane space and a low concentration in the matrix. Hydrogen ions are going to be using the process of diffusion. Link up in the top right if you need a review over that. And this movement of hydrogen ions is going to be captured by this F0 complex, which is the rotor. Okay, so the F0 complex of ATP synthase is the rotor. So let's get a little different angle here and take a look at the rotor of ATP synthase. So hydrogen ions are going to enter through a channel which has not been fully molecularly characterized, but what we do know is that these hydrogen ions are going to be driving the rotor. And so we're going to be using mechanical force to turn the rotor. And so during this process, the hydrogen ions are going to be moving from the intermembrane space, which is the higher concentration, out to the matrix, or into the matrix, I should say. And so that movement, that energy of from high concentration to low concentration, is going to be powering this mechanical rotor, kind of like a water wheel or a windmill. So that windmill, that F0 complex, is pushing energy into the F1 complex, which is this mushroom head hexamer with the blue and pink proteins. And so that's going to be changing the conformation of these proteins, which allows ADP to slip between the blue and pink proteins here, and inorganic phosphate, acronymed PI. And so that's going to allow us to synthesize ATP. And so ATP is synthesized from this process. So getting a little closer look here, zooming in between these complexes, take our inorganic phosphate and we're going to bind that with the ADP using the conformational changes created by chemiosmosis in the rotor. And that creates our ATP molecule, or with our 3 phosphates there. And so we're not going to worry too much about the numbers. However, for those of you that do need to know these for your test, we're going to be creating 28 through, so ATP synthase is going to create about 28 through 32 ATP. And so different authors of different books are going to be using different numbers, and that depends if they take into account some ATP that's used to funnel in pyruvate, or the hydrogen ions that funnel in pyruvate. Also, sometimes in some of our cells we have some differences in NADHs that are coming from glycolysis. Those sometimes are funneled in by way of giving electrons to FADH. So whatever your textbook says to remember for electron transport chain, remember that. So for all of aerobic respiration, our net output per glucose is anywhere from 32 to 38 ATP, depending on your textbook. 6 CO2 molecules, which for mammals and those with lungs, we're going to be breathing that CO2 out. And 6 H2O, which our cells usually utilize and we keep. But again, it is mostly a waste product. So that does it for the electron transport chain. That was a lot of information, so I encourage you guys to watch some steps a little bit slower, maybe turn the audio off and try to explain it yourself, because that's really going to help you to learn this material. And so that does it for this time. I hope to see you guys in the next video. Appreciate you watching. If you liked the video, please subscribe. If you learned something, please give me a like and subscribe. I really appreciate it, guys. Thanks.